Our original sequence from the 5' end to the 3' end is ATG, TGG, AAC, GCT, TGC, TGA, CCC, and GGC. When translated, this is AUG, UGG, AAC, GCU, UGC, UGA, CCC, and GGC. Next, we find the amino acids that correspond with these codons. They are methionine, tryptophane, asparagine, alanine, cysteine, and then stop. In this strand, we ignore the last two codons since the sequence stops before it reaches them. Like with most things in genetics though, many things can go wrong. In this video, we'll focus primarily on point mutations, which is when a single nucleotide in a codon is altered. There are four major types known as missenses, readthroughs, nonsense point mutations, and silent point mutations. The first, a missense, is when there is a change in the amino acid that also causes a change in the protein, specifically its function. With our original strain, let's say that on the 5th codon, a transversion mutation occurs and the U changes into an A. This would change both the DNA and the mRNA codon from UGC to AGC. UGC codes for cysteine, while AGC codes for serine, thus changing the protein function and making this a missense mutation. The next is a readthrough, which is when a stop codon is mutated into a regular amino acid, and the protein chain becomes longer than before. In our example, our stop codon is the 6th in the sequence. If the A were to transition into a G, that would change the stop codon into tryptophane instead, and the sequence continues on. This is where we would actually include the CCC and the GGC, coding for proline and glycine respectively. Since there is no stop codon, the sequence continues on until there is another place to stop. The third type is known as a nonsense point mutation, which is sort of the opposite of a read-through in that a codon for an amino acid is mutated into a stop codon. In our example, if we look at the second codon of the sequence, a transition mutation from G to A would change the UGG, which normally codes for tryptophane, into UGA, which is a stop codon. The final type is known as a silent point mutation, and this is when an amino acid is changed, but it still codes for the same protein as the original. If we look at the third codon in our example, if C undergoes a transversion mutation into U, thus changing the codon from AAC to AAU, then we can see that it will still code for the amino acid asparagine.